Hello everybody, this is still chapter 21, picking up with slide 61. And this is where things are going to get really down into the nitty-gritty of exchange. So what is happening at the capillary level? This is kind of the entire purpose of the cardiovascular system. So we are delivering the products. We're delivering oxygen picked up from the lungs, and we're going to be delivering nutrients that we will pick up from the liver. And we have to deliver them to every tissue in the body. Okay? So in order for cells to stay alive and maintain homeostasis, they need oxygen, they need nutrients, and they need wastes removed, including carbon dioxide. And so we're going to be moving things through the capillary walls into the interstitial fluid, and we're going to have three processes that are happening. Diffusion. And remember, in diffusion, things go from where they're highly concentrated to where they are less highly concentrated. We're going to have filtration. Filtration is based on a pressure difference. So whichever side is exerting more pressure is going to push things through to the other side. And then we have reabsorption. Now, reabsorption is kind of related to diffusion, but it's more about the osmosis part. So if I push a bunch of fluid out of a capillary, but I trap the big plasma proteins in the capillary, those plasma proteins attract water backwards back into the capillary. So we squeeze it out and then we suck it back in. So these three processes are all happening at the same time in the capillary beds. So again, diffusion is movement of ions or molecules from where they are highly concentrated to lower concentrated, and they follow their concentration gradient. The cool thing about diffusion is it doesn't cost us any energy. It is the natural tendency of things to move from where they are more crowded to where they are less crowded. So if I bring blood into the capillary bed that has a high concentration of oxygen, and the interstitial fluid has a low concentration of oxygen, then that oxygen wants to move from the blood into the interstitial fluid naturally, just by the process of diffusion. It will not have to be pumped or physically moved over there. Okay? So, diffusion routes. Which is going which way? Water, ions, and small molecules like oxygen can diffuse between the endothelial cells or through pores in the capillaries. Some ions have special channels for diffusion, like sodium, potassium, calcium, and chloride ions. Large compounds that are water soluble can only get through fenestrated capillaries, those ones that had pores, right? Lipids and lipid-soluble materials such as O2 can go right through plasma membranes, and plasma proteins can only come through the lining of sinusoids. Otherwise, they are trapped inside the capillary. So this is going to be really important as we're deciding what is able to move in and out in each of the types of capillaries. So again, our second force is filtration. It is by a difference in pressure. Now we're calling it hydrostatic pressure because we are talking about fluids here. One fluid is blood, the other fluid is interstitial fluid. Blood has heart behind it. It has been squeezed and pumped, and there is a certain amount of blood pressure when the blood arrives into the capillary. So if the pressure pushing in the capillary is higher than the pressure exerted by the fluid outside of the capillary, things are going to move out of the capillary into the interstitial fluid. So because of the hydrostatic pressure from the heartbeat, water and small solutes are going to go through the capillary wall and be outside of the vessel. But this leaves larger things, larger proteins and solutes still in the bloodstream. And it's these larger solutes and proteins in the bloodstream that lead to our third force, which is reabsorption. It is as a result of osmosis. Now remember, osmosis is the diffusion of water from where it is less concentrated or more highly concentrated to where it is less concentrated. So if I have a lot of water that is driven out of a capillary by a heartbeat, and then I have a lot of protein and solutes still in the capillary, the water is going to go back in. Now, the amount of pressure exerted by the proteins in the blood to draw water back in reabsorption has this lovely name, the blood colloid osmotic pressure. So that's the big proteins that are in the capillary that can't get out, that draw water back in. Okay, so again, capillary hydrostatic pressure, or CHP, is leftover pressure from the heart beating that pushes things out of the capillary into the interstitial space. So this drives things out. And here's an up close view, right? And it goes right between the cells or through channels or sometimes through membranes. So what's going on between filtration and reabsorption? Having both ensures that plasma and interstitial fluid are in constant communication, and we're always having exchange of materials. It helps to accelerate distribution of nutrients, hormones, and dissolved gases. Helps by bringing insoluble lipids and tissue proteins that cannot go through capillary walls. And carries the toxins from bacteria and other chemical stimuli to lymphatic tissues and organs. Filtration and reabsorption are affected by the differences between the hydrostatic pressure and the colloid osmotic pressure. So notice these say net. So if they say net, this is after we subtract something from something else, right? What we're left with. So factors that contribute to net capillary hydrostatic pressure, remember this is from the heartbeat, is the hydrostatic pressure from the capillary minus the interstitial fluid has its own liquid pressure. But this one usually tends to be higher than this one, so this overall tends to push water and solutes out of the capillaries and into the interstitial fluid. Now let's look at blood colloid osmotic pressure. There are proteins that are outside the blood vessel. That's called the interstitial fluid colloid osmotic pressure. And then we have the proteins that are stuck in the blood. Overall, usually this one, number 1, is bigger than number 2. And so the net result is we're going to pull water and solutes back into the capillary from the interstitial fluid. Okay, so we push a lot of things out, we suck some water back in, and then the blood goes into the venules. So net filtration pressure is the difference between the net hydrostatic pressure and the net osmotic pressure. It's a lot of math, isn't it? I have good news for you. I'm not going to actually make you calculate it, okay? Just understand the concepts and I will be happy. So what's going on? On the arterial end, so right after the arteriole, we tend to have more of the fluid leaving the capillary into the interstitial fluid because of the leftover pressure from the heart. Okay? So on one end of the capillary bed, we're driving fluids out of the capillary. And we're delivering oxygen, and we're taking out nutrients. So as all that stuff is being pushed out by pressure through filtration, they are entering the interstitial fluid. Okay, the blood is still moving forward through the capillary bed. By the time it gets to the venous end of the capillary bed, it has pushed out a lot of fluid and has trapped the larger proteins. And so on the venous side of a capillary bed is where the reabsorption happens. Okay, so we're sucking back in some of that fluid we just pushed out. The transition point is closer to the venous end than the arterial end, so we end up having less time for reabsorption than we did for pushing things out through filtration. That means we don't get to suck all that same fluid back in. So capillaries filter or push out more than they reabsorb, and so fluid is building up outside of capillaries. Luckily for us, we have a second circulatory system called the lymphatic system, which we don't get to really talk about in this class. I'm very sad. But there are lymphatic vessels in the same tissues that pick up this excess fluid that did not get picked back up by the capillaries, takes it through lymphatic organs, examines it for pathogens, filters it through lymph nodes, and then puts it back into a vein up near the heart. So we're going to add that fluid back in so we don't have a drop in blood pressure because we took all the fluid out. But it gives us another secondary way of collecting the fluid and monitoring it for infection and filtering it. So this is a key diagram for this chapter. If you can understand this diagram, you will do well on this test, okay? Again, you don't have to memorize the numbers. You just have to understand what's going on. So here is our arterial end. As the blood is coming in here and enters the capillary, it still has pressure from the heartbeat that pushes out fluid of about 35 millimeters of mercury outward, but it's resisted by the fluid out here by 25 millimeters of mercury. So what's 35 minus 25? 10. So our net filtration pressure on this end is 10 millimeters of mercury pushing fluid out. Okay, so filtration wins. And filtration from the capillary to the interstitial fluid. As we move this blood over here, we get to a point where they're even. So we actually have the same amount going out as having come in. And as we move to the venule end, we actually have less pressure now left over from the heartbeat. It drops from 35 to 18, whereas we still have the 25 millimeters of pressure coming inward. So now we have more coming in than going out. And so we actually absorb stuff because of the negative 7 millimeters of mercury of pressure. So filtration happens over here. Reabsorption happens over here. And if you look, you see the difference between the two. So we actually leave a lot of fluid behind in these interstitial spaces. I hope you understand that. So that leftover ends up being about 3.6 liters a day, gets picked up by these lymphatic vessels and returned to the circulation. So you push out 24 liters a day in filtration, you only reabsorb 20.4 liters per day in a reabsorption, and that other 3.6 goes into the lymphatic system. If you start to bleed and hemorrhage, that messes this all up, because it reduces the capillary hydrostatic pressure and net filtration pressure. So what happens is you end up increasing reabsorption of interstitial fluid, called recall of fluids, because your blood pressure has dropped. It ends up doing more reabsorption than filtration. Dehydration also increases blood colloid osmotic pressure, which accelerates reabsorption. So we have two reasons why absorption is going to win over filtration if you're bleeding. And if your capillary hydrostatic pressure rises or your blood colloid osmotic pressure declines, then your fluid moves out of your blood and builds up in your tissues, known as edema or swelling. Okay, so tissue perfusion. I told you one of the main purposes of this capillary bed is to make sure we have enough oxygen in our tissue. And we take away CO2. CO2 is a waste product that is toxic to cells. So tissue perfusion is making sure we have enough blood flow through the tissues to deliver enough oxygen and nutrients and to efficiently remove CO2 and waste. And it is also affected by cardiac output, peripheral resistance, and blood pressure. When certain cells become active, circulation to that area must increase. So we must have a way to change how fast and where blood is going. So that is what cardiovascular regulation does. Ensures that the blood flow changes occur at an appropriate time, in the right area, and without changing the blood pressure and blood flow to vital organs. Okay, so this is going to start to get deep into this. I'm going to stop this presentation at 76, and I will pick up the next one at 77.